Hi everyone, this is Heather Benson and I'm going to be explaining how to work through the problems for the Bio 375 Exam 1. So I think it'll be helpful for me to show my work, how I did this. So I'm going to share my screen for the rest of the video here so we can work through the steps. Okay, so for question one is concerning the test done with purple flowers and long pollen plants and red flowers and round pollen and all of those traits. So the first thing that we do to work through this problem is you have to propose a null hypothesis to use in your chi-square analysis. So for this question, I propose that purple is dominant to red and long is dominant to round, and that flower color and pollen shape do follow Mendel's laws of independent assortment and segregation. Then here we have the parents, the original parents. When those are crossed, they produce this heterozygote cross, and after that you take the heterozygotes and you cross them together, and this is used in the F2 cross to produce this F2 generation. So then we have this ratio that we get, and it is a 9:3 to 3:1 ratio with, as you can see here, purple flowers, long pollen, purple flowers, round pollen, red flowers, long pollen, red flowers, round pollen. So what you do here is you total up what they put up here, that's 320 total plants, and you multiply that by the ratios you got, and you fill out this table here. So here we have the observed values. Those are the number of plants that were given above. And then here we have the ones that we've multiplied out over here for what we will expect. Then for the chi-square test, we take the chi-square value and we do observed minus expected squared over expected, and you do that with each of these crosses, and you can just use your table here so it works out really simply. Then after you multiply all that out, you get 168.53 for the chi-square value. The degrees of freedom for this problem is 3 because there are 4 phenotypes and you just subtract 1 to get the 3. So when we look at our chart up here, you notice that 168 is much higher than what our null hypothesis, or what our chi-square value should be for degrees of freedom of 3. See, it's much higher. It's definitely in the gray zone. So because of that, we have to reject the null hypothesis. That means that flower color and pollen shape do not follow Mendel's laws of independent assortment and segregation. The new null hypothesis that I would propose is that flower color and pollen shape don't follow Mendel's above stated laws, and that a gene interaction is occurring, such as epistasis. So moving on to problem number 2. This problem concerns Huntington disease. For this question, they want to know the probability of what they will get with in their children based on different crosses. So what is the probability that all 6 of their children will be affected with Huntington disease? So we have a heterozygous female who marries a unaffected male. So we show that here. And this is their progeny. So phenotypes here would be half diseased, half non-diseased. So if you want to know if all 6 of their children will have Huntington disease, you need to multiply 1/2 times 1/2 times 1/2, do that 6 times because they're independent events, and you'll get 1.56%. For part B, what is the probability that 4 children will be unaffected and 2 children will be affected with Huntington disease? The probability of that, what you have to use here is this equation here. And excuse me. So for this number here, we have our probability. And this is the total number of children. So that is 6 children because we have 4 children and 2 children. And then for here, we have 4 children. That's what we're looking at. 4 children will be unaffected. And this is factorial, which basically means 4 times 3 times 2 times 1. And this is multiplied by 6 minus 4 factorial, which is 2 factorial. And you multiply that out, multiplied by 1/2 to the 4th power, multiplied by 1/2 to the 2nd power, and you get 0.2344, or 23.44% of the children. Or what is that is the probability that the children will be 4 children unaffected and 2 children affected. What is the probability that the first 3 children will be either all affected with Huntington disease or all unaffected? So what we do here, we know that it's 50% diseased or 50% non-diseased. So you independently calculate that the first 3 children, either all affected, or, that's what the plus sign means, or all unaffected. And you get 0.25 when you multiply that out, which is 25%. Okay, so for problem number 3, this has to do with the mode of inheritance for these diagrams here. So this one is X-linked dominant, and the key factors in determining that have to do with that all affected children have an affected parent. You can see this doesn't skip any generations. The daughters of affected fathers are all affected. So we have an affected father. All of the daughters here are affected. Both of these are. For part B, it's X-linked dominant, and that's because males and females are affected. Every affected child has an affected parent. The only shown affected father has an only daughter who is affected. For part C, it's X-linked recessive, and that's because affected children don't have any affected parents. There are all males and no females affected, and also the shown affected father's son is not affected. That's shown here. For part D, it's X-linked dominant. Both males and females are affected. All affected children have a parent who is affected. And the shown father's daughters are each affected. Okay, so for problem number 4, this is about the one about the African hedgehogs and their chromosomes, etc. So for part A, how many sets of chromosomes are present per somatic cell in this organism? That would be 45 sets. That's because there are 90 total chromosomes divided by 2 equals 45 sets of chromosomes. How many tetrads are found per cell during prophase 1? Well, that would be 45 tetrads because there are 90 homologous chromosome pairs and they're lined up, which causes this. How many sister chromatids are found per cell during metaphase 2? That would be 90 sister chromatids. That's because they have not yet been split, but that comes next during anaphase 2. For part D, how many chromosomes are found per cell during prophase 2? That would be 45 chromosomes, and that is half as many as would be in prophase 1 because the division has occurred. So for number 5, the last one here, this is the tetrahybrid cross in pea plants. And so be sure to read through all this here. So what is the probability that a dwarf wrinkled yellow terminal offspring plant is produced? And what you do here is, so I did the Punnett squares up here so that you can be sure you don't get anything confused. So I set it out like this: dwarf, 1/4, wrinkled, 1/4, yellow, 1/2 up here, terminal, 1/2. Then you multiply that. 1.56%. Part B, what is the probability that an offspring plant has this genotype? So again, I do the same thing there. Write it out, 1/2 TT, or heterozygous, 1/2 heterozygous, 1/2 heterozygous, 1/2 heterozygous, 6.25%. Part C, what is the probability that the first 5 offspring will have this genotype? So for this one, you do the same sort of thing. 1/4 homozygous dominant, 1/4 homozygous recessive, 1/2 heterozygous, 1/2 homozygous recessive, and you get this number here. And if you want to know the probability that the first 5 offspring will have that genotype, what you do is raise that to the power of 5. So that's multiplying it by itself 5 times. You get this here, and you multiply that by 100 and get 9.31 times 10 to the negative 8% of these offspring. That's the probability that the first 5 offspring will have the stated genotype. For part D, what is the probability that an offspring plant will have the dwarf round yellow axial phenotype or the dwarf wrinkled green terminal phenotype? So what you do is you calculate these separately. So we have dwarf round yellow axial, do the same thing as above. We have dwarf 1/4, round 3/4, because that includes the heterozygous and the homozygous dominant, yellow 1/2, axial 1/2. You calculate that, then you do dwarf 1/4, wrinkled 1/4, green 1/2, right here 1/2, terminal 1/2. Then you multiply those together and you get this number here. Now, because they say, what is the probability of getting this or means that you add them together and you get this number here. You multiply that by 100, and that turns out to be a probability of 6.25%. So I hope that that made sense, and thanks for listening.